Hello everyone, welcome to Get My Class. So let's solve this question. The question says, which of the following statement or statements is or are true? Option A says Sanger is credited for developing method for determination of amino acid sequences in proteins. Option B says the fragments were sequenced using automated DNA sequencers were not based on Sanger sequencing. Option C says DNA sequencing worked on the principle of a method developed by Frederick Sanger. Option D says both A and C. What does the question say? The question says which of the following statement or statements is or are true. So we have to look for the true statement or statements. So let us see one by one. The first one says Sanger is credited for developing method for determination of amino acid sequence in protein. So let us talk about the first statement, which is Sanger. So who's Sanger? Actually, they are talking about Frederick Sanger. Okay, Frederick Sanger. Okay, Frederick Sanger was a scientist who developed methods to sequence nucleotides as well as amino acids. Methods to sequence nucleotides in DNA, even you could say DNA or even RNA, as well as amino acid or amino acids in proteins. Okay, we all know that proteins are made up of what? Their proteins are made up of amino acids. So they have particular number of amino acids in each type of proteins, right? And even the type of amino acid will differ. Okay, so we have totally how many amino acids? We have 20 amino acids. 20 of them we have. Okay, but then the arrangement of the amino acid in the proteins or in the polypeptide chain can vary. Even if you talk about nucleotides, how many nucleotides there are 4 different types of nucleotides, right? ATGC, if you talk about DNA, ATGC, and similarly AUGC in case of RNA. Okay, so these are the 4 nucleotides, but the way they are arranged in one big stretch of DNA, it differs, right? It is not that it is in the same format they'll be present. It can be like AAATGC or AATTGGCC, some, depending upon the nature of the particular sequence, you can actually get different ways of arrangement of the sequence. Okay, so that is why like Sanger, that is Frederick Sanger, he developed a method of sequencing the nucleotides present in a particular nucleic acid, as well as sequencing the types of amino acids present in proteins. Okay, so that will help us to understand, at which sequence are responsible for what characteristics, and which type of amino acid sequence will result in what type of protein. Okay, so option A, it is a true statement. So that is why he was credited for his method of developing, or you could say for the development of the method for determination of amino acid sequence in protein. That is absolutely right. Option B says fragments were sequenced using automated DNA sequence were not based on Sanger sequencing, which is incorrect. Because once Sanger developed the sequencing method, the same method was used for automated DNA sequencers. Okay, so his methods were used for automated sequencing, you can say. Okay, automated sequencing, that is nothing but using machines. Okay, so this information has been fed. Okay, the information given by Frederick has been fed in these automated machines, and these machines will use the same principle and try to sequence the nucleotides in the DNA, or you could say, since they are talking about DNA sequencers, so that means they are talking about sequencing of the nucleotides present in DNA. Then DNA sequencing worked on the principle of a method developed by Frederick Sanger. This is also absolutely right. And option D says both B and A and C. That is the perfect answer because it includes both A and C. So A, which says Sanger is credited for developing this method for the determination of amino acid sequence, that is also correct. And C, which says DNA sequencing worked on the principle of a method developed by Frederick Sanger, that is also correct. So we will go by option D. Okay, so what is the answer? The answer is option D, where both A and C are involved. I hope you understood the explanation. Thank you, everyone.